Well, good day, everyone. I am Ross Caputo. I'm President and CEO of Eagle Analytical, and I'm also a microbiologist. I am here today to talk to you about mindset of quality compound, but first things first, I have no conflicts of interest. So with that, let's get started. The learning objectives today, what I wanted to share with everybody is I wanted to talk a bit about mindset, which is a very vogue concept, but the mindset of quality compounding is a little bit different. I want everyone to understand the I want you to outline the right things to do so that compounders need to adopt in order to avoid lagging behind the industry and where it's going, where it's shifting. Now we're going to discuss the differences between a fixed mindset and a growth mindset as it relates to our pharmaceutical compounding and patient safety. We have got to demonstrate a comparison, and I will help demonstrate a comparison with you to these alternative mindsets so that you don't get caught up in between and thinking this is the right way to do something or it's not the right way to do something and what's going on around me. Because let's face it, we have all had a change in mindset due to COVID. The pandemic has changed everything in how we look at things, how we do things, what we care about, is begin to focus us truly on value and the means to attain it. Now, this is sort of a little picture for everybody to see. A quality budget before and after getting a warning letter or a nastygram from the boy to pharmacy, as you can see, it appears that we get religion right after somebody calls us on it. There is no doubt that it costs a lot of money to be a non-conformance. It's a lot cheaper, a lot more value added if in fact you invest in the appropriate things that you need to have the right quality attributes, the right mindset of how you should approach patient safety, regulatory compliance, and the value of your business. Now, I want you to contemplate a bit about mindsets. Now, I've given a reference here, and I'll show it again. Dr. Carol Dweck, who wrote the book. The book has had several printings. There's 2.5 million copies out in the world. She's a psychologist, and her intention is to teach people, or enlighten people, perhaps is a better word of saying, how you already have in your mind a set direction that you will embrace. A fixed mindset, and boy, we know what a fixed mindset is. A fixed mindset in our children says something like, well, you know what? I'm not going to take that class because I don't think I can get an A in it. And an A is more important to me than knowledge. And I'm as good as I'm going to be today or any other day. So let me take that easier road out because I'll feel better about myself. This person is saying, we personally need to prove that we are correct and that there's really no room for fundamental learning. You're either a great athlete or you're not a great athlete. You're either good at math or you're bad at math. You either get it or you don't. A growth mindset on the other side, however, believes that your basic qualities are things that you can achieve and cultivate and grow through efforts, strategies, and help from others. There are many topics, many books that talk about being a continual learner, to embrace change with progress. But you know what we're really talking about there is what is your process of how you approach each and every day, each and every question, each and every opportunity. We have a lot of words that talk about nothing ventured, nothing gained. We talk a lot about things as endpoints, but a growth mindset is a process. A growth mindset is not a destination. It's not a proclamation. It's how do we work through what we're doing when we do it. Just let that settle in for a minute. You know what we're talking about. It's a little fuzzy every now and then. So we're going to try to use some perhaps funny, but so very obvious demonstrations and examples. Now, I'm not saying what profession this gentleman is in wearing the bag. He could be a sports fan who doesn't like his team any longer. But at the end of the day, the person is saying, I've done this for years without an issue. I interpret the rules differently. I have professional judgment. And if I am questioned, I must be professionally incompetent. Therefore, I must absolutely fight to the death that I'm right no matter what, and I'm going to stick my feet in and I'm going to go do that. That is truly a fixed mindset. It is truly an issue that actually doesn't give you much hope for growth or even how to understand what's going on. But what happens? Well, if you have a growth mindset, and I want to talk about a few things that we heard this morning. Gail was talking about the emergency use approvals that were given out. That was a growth mindset, wasn't it? Even though it's stuck in there to do that. So look, whatever these rules used to be, there's something different going on now. And our process says we have to do things differently. We have to identify what opportunities we need to go after, and we need to figure out how to do it. We need to execute it, but we need to stay on top of it and review that the changes and the process activities that we were making made sense. That is a big deal. It made sense. Now, a fixed mindset says, oh, well, you changed them once. The heck with it. I don't ever have to go back and do those things again. I'm going to keep doing what I was doing, because when it came down to the brass tacks, you BOP or you FDA or you anybody you said, I didn't really have to do it. I didn't really have to get that done. As opposed to understanding that a growth mindset studies a process, studies a reaction, studies a value treatment. We are in dire straits with this pandemic. We were in dire straits not knowing about what drugs we could use, what vaccinations could get done, how could we get it out there. But the growth mindset said, let's figure it out. Think of that situation as being a classic example of the management of benefit risk decision making. We needed to do something. We needed to do something and watch us make a process that shortcuts but doesn't alleviate requirements to get to point A to point B quicker, but then to go back and make sure everything is covered once we're out there. Benefit risk, growth mindsets, opening up to a process. I'm going to use many times here, process versus proclamation. I think therefore I am was a very famous statement, but that doesn't work. Just because I think about it doesn't mean it's right, right? What it says is, why am I right? What are my metrics? What do I do with it? Many times when we get 483s or accusations or questions from a board, are immediately say, well, wait a minute. I've done that. I know what I'm doing. I don't need to do that. We really need to get into the mode of taking those very pointed observations and suggestions and think about what they need. Pharmacists and compounders and producers know more about their process than any inspector that walks in the door. Pure and simple. You know more about it. They are asking questions. We ask questions with a growth mindset to see, is there more we could learn or should learn? How do we do this? What is supposed to happen? A continual growth mode. I like to think of it, if I get smarter at it, I can do it more efficiently, and frankly, you're in business as well, right? And more profitable. So when I go look at some mindsets in compounding, and I want you to know that please consider mindsets in your everyday life, from cooking to cutting the grass, to talking to your children, to going to the school and figuring out what should be going on or not going on. Think of everything as a process of evaluation with metrics leading the way. But leading the way means you need to know where you're going. You need to understand what the endpoint is. So we're going to use a couple examples here, some of my favorite ones. Media fills. Remember, I'm a microbiologist. Everything lives and dies by bacteria and viruses, etc. So I'm all over that. But when I read 797, I must read it different. 797 says you need to take 9 vials and go do a basic approach, and this is what it is. And to me, I always thought what that meant was how I teach the pharmacy tech or the new person I just hired to frankly see if, like me, they're all thumbs. See, I'm not allowed in my lab. These hands don't work as well as they used to. And actually, dexterity and control are very important in aseptic operations, isn't it? So I look at 797 as how to figure out in general if someone can do it and sort of your basic training in the first place. But a fixed mindset, and you can see it in Board of Pharmacy's reports, you can see it in 483s. Their response is, that's all I have to do, that's good enough, that's all my Board of Pharmacy is required, and I've not got written up for it. Well, I think you heard from Gail, and I think you can read between the lines of all the other approaches that came through. USP requirements, first of all, are always minimum, right? And the Board of Pharmacies, they focus on the regulations in and around the regulatory compliance for patient care and safety of it's the right script, it's the right drug, it's the right dosage form, it's being done the correct way, the drugs are not being confused, they're not going to the right places or wrong places. There has been a shift, a paradigm shift since 2012 that said process is important when you're making a sterile product. It was newer news, if you will, for the world of pharmacy, not for the world of drug manufacturing, aseptic processing in a global way. It wasn't new. When we look at growth media fills, you need to open up your mind and say it's a holistic approach. What do I mean by that? 797 gives you a hint, but frankly doesn't totally define it. It says you have to simulate the most challenging of conditions, right? Or stressful conditions. Do you know that assumes that you actually know what they are? And I'm real sure that I know many more and many different as a microbiologist, as a person with GMP experience, than a pharmacist does who has been making these products for maybe 15 or 20 years. I couldn't compound a drug product, get it out with the right labeling, the right dosage, and all that kind of stuff. I wouldn't even attempt it because I don't know it. That is not my world. But I do know that in growth mindsets for media fills, you have to not only mimic the process, it has to absolutely be the process in every step that could impact an aseptic manipulation to make it more difficult. And what I mean by that is increase the ability to cause contamination. If I were to make an aseptic manipulation in an ISO 5 hood versus the same aseptic manipulation with the same person in the parking lot, you know those things are apples and oranges difference. Even though it's the same person with the same manipulation, because it's in a different environment, it poses a different risk. It poses a different threat. Growth mindsets, what in this particular case, are saying, think about it. What does it mean? Does it have an impact? I put on this slide, it says, it mimics the process. You have to have a dynamic condition. Dynamic condition means people that could be in the room at the time doing whatever they may or may not be doing, so as to cause a turbulent flow, to see if those air changes per hour are going to take care of the room, if your manipulations are correct under the hood, making sure that they get done, to make sure that it happens correctly, that I'm dressed appropriately, that it was a hot day and I'm sweating, but all my skin is covered, that I have sterile garb on where I should have sterile garb. Now, as a microbiologist, I have always been confused about the concept of being able to wear non-sterile anything in the hood environment. We have to spray down everything before it goes in the hood. Everybody knows I got to spray down with alcohol, I decontaminate it, I got to do A, B, C, D. And yet for a while, and even today, frankly, until new things come out, you're allowed to put arms into a hood. And I keep asking if you spray them. I know it's a gown. I know it's non-sterile. Some people use sterile sleeves. Well, In my mind, when I'm doing my media fills and I've got my growth set, I'm evaluating what I'm doing. I see the guidance as suggestions and recommendations of what I should know. I don't take it as the textbook to say it's the final answer, because I'm going to be doing a whole lot of different things. Some may be doing sterile to sterile transfer. Some may be starting from 3 or 4 different high-risk products, non-sterile, that have to be sterilized, have to be mixed, that have to be put together to go into my final dosage form. Apples and oranges, very different. When we're looking at media fills, we're looking at microbiology. So I need to know air, contamination, as well as cleanliness. I need to know surface contamination, as well as cleanliness, and the same thing for personnel. We know, we've read, so let me put it out there. Some people can't go in the clean room. They're shedders. Period. End of discussion. They're shedders. Well-known fact. You can look it up. You can Google it. As Batman says, that's worth a Google. Go look it up. It's there. But if we have a fixed mindset, we believe that all people are fine. All people can go in, and if I teach them what they can do, they won't impact the quality of my product. And that's not necessarily true. If I'm making 4 or 5 eaches in a batch, not a problem. If I'm making several hundred, what does that tell you? I'm there for a lot longer time. I'm stressed. I'm activities. I'm making work. I'm causing all these little, go back to the old Snoopy cartoons where Pigpen's got this flood of dirt around him and he's walking around. That is a fact. It's a reality. A growth mindset just thinks about it. Doesn't mean you have to act on it. It doesn't mean that you have to say, oh my God, I've got to do all these things different. No, it doesn't mean that at all. It's a process. A growth mindset says, let me find out what I'm doing. When we talked about that, we talked about environmental monitoring there, didn't we? Well, what do we have on our fixed mindset? We're back to nobody wrote me up, so it must be okay, right? Everything is fine. 797 say you do plates every 6 months. So they say you do active air for fingertips. They do that you do active air samples every 6 months, and you should do USP 797 says you should do surface sampling, but doesn't give you a frequency, does it? Well, I could just tell you that nothing on this page made sense to me, because remember, I'm a microbiologist. And I know that if I only take plates every 6 months, I have no idea what's going on any other time. I can't do a process control chart. I can't see my trends, because those are the kind of things that in a growth mindset, I'm going to ask those questions. I want to know if I'm in control or not. I want to know what I should be thinking about. If I have one hood in one room that is an ISO 5 with ISO 7s, then with an ISO 8 total control, and I'm making 4 dosage forms a week, very different for me on what I'm concerned and worried about. But if I have multiple hoods in multiple places, making multiple 20, 30, 40 and above scripts a day of different compounds and different drugs, so that means you're going in and out and in and out. There is a lot of activity going on. Please remember about environmental monitoring and personnel monitoring. No self-respecting bacteria flies. It is carried to a specific destination. And what carries it? Hello, we do. And how do we do that? Our bodies, our touching, where we were supposed to wipe down or disinfect something, maybe we didn't get it quite right. We have to carry it in. We have to bring it in there. So that means that we need to be able to demonstrate a state of control. We have to think about a lot of activities. The in and outs, mean every in and out that you have an opportunity. Remember that dynamic testing you're supposed to do for media fill? You're supposed to do that with people purposely going, well, they go in out 3 or 4 times. And yeah, maybe sometimes 5 times. I go in there every now and then, I go look at it. But it seems to me that every time that I see a warning letter or an activity, it says, well, just this one time, the person went from the hood out through the control room because he forgot something on his shelf, and he came back in and he didn't disinfected or didn't change his gloves properly. You can look that one up. And I look at that and said, that's all they pointed out. There are like 9 things wrong with that whole statement, right? So a growth mindset, and I just want you to think about things. I'm not telling you to do anything crazy. I'm telling you to think about the process of evaluating the risk and then the benefit of having the right metrics in place to get things done before it becomes a problem. It is everybody's job to learn what's going on in anything they do, but especially in the production of drugs. Sterile or non-sterile is irrelevant to me. Non-sterile doesn't mean dirty, right? So keep all of these things in mind, even when we're talking here about sterile products. Batch size matters, trend analysis or requirements. You cannot trend 2 points a year. That's silly. I understand new rules are going to be coming out. And for those of you that are just simply going to do the minimum, well, they're going to increase the minimum for you. Those of you that are going to take and look at your process to find out if some particular processes require more, and frankly, others may require less, please don't go overboard, but by God, understand the value proposition and what you need to do. You should trend everything. You should process everything. You should understand what's going on. And please remember that batch size matters. So, got to talk a little more specific about a point that for a gentleman like myself, coming from the pharmaceutical training, interesting in stability, and a lot of the discussion about beyond use dating and potency over time versus stability indicating assay, I actually get both sides of the coin. Potency over time is a great idea because that is what a stability test is, if and only if the method that you're using can give you the information you're seeking. Now, I know the fixed mindsets today because I see it all the time, and I understand the frustration. I understand the frustration is I've been doing this for years. I haven't changed anything. It works for me, and my professional judgment says it's okay. Now, here's the good and the bad about that. It probably is okay. Most of the time. It's the rest of the time that's the issue. And let's face it, from 30 years ago to today, expiry dates and BU dates have gotten longer. And the longer it sits, the more you are apt to have an issue, and you have to predict that issue when you can. And it's not like falling off. It's not like falling off a cliff. What it's really about is how do you predict it when it's going forward? Now, I'm going to tell you something. In fact, I was going to put it on the slide, but I didn't, but I'll read something to you. Stability indicating assays, the first published production of requirements for it was in 1994. And they had been working on that for several years. It was revised in August 2001. And it was put out by, ICH. Some people have heard of ICH. Well, that's the International Conference on Harmonization of Technical Requirements for the Registration of Pharmaceuticals for Human Use. Okay? And that includes harmonization of drugs and drug products, and specifically this ICH Q1A, we're now in revision to R2, is all about stability testing. It's been harmonized such that Europe, the European Union, Japan, and the US all agree. It's a harmonized document. This is how you do it. Please, I just told you about something that's nearly 30 years old. So it doesn't sit well with our discussions in the compounding world to say, what is this new stuff? It's not new. It's being applied now. Two different things. So please understand the application of it is what's new, but not the science. Now, I tell you something else. When you see this, and you see these questions of degradation, accuracy, linearity, specificity, specificity, precision, and detection, all of those are set studies that must be done as you degrade these products on multiple modes of degradation. Yes, it's true. Pharma products can be out there 2, 3, 4 years. But some compounders are now having products out there 6 months, a year plus, etc. And by the way, there's always degradation. The question is how much, right? Not if any. The question is how much. And we can measure that in the potency. And usually, now here, listen to me carefully, usually what happens, because these products have been used for a long time, we don't see or haven't seen any issues of toxicity or issues or problem that we're aware of. We can suggest, but not know, suggest that the breakdown products are not toxic, that they don't cause an issue, and they don't interfere with anything. That's what this is about. What it's about is making sure that breakdown products, that the drug still must be effective, but the breakdown products are not toxic. Breakdown products are not interfering with the therapeutic treatment. Again, this is all about growth mindset of stability is know what you're doing, know what you're saying, and when you get it done. And please remember, stability indicating assays and studies, which there's all sorts of methods that are approved, bracketing and matricing, et cetera, to get down to a fixed number, if you will, that you do one time. Now, you're constantly going to take a look. If you change this or you change that, you go in, you run a study, you take a look at it. But most don't change a whole lot. Most do what they're supposed to do, right? You buy good quality materials from APIs from FDA registered facilities. They're part of USP, they're NF, all the stuff that you're supposed to do. And as such, you have every reason to believe that your product is fine, that your product will be safe. And then this test proves that it is. That's what this is about. Sterility test. Method suitability has, in a fixed mindset, says, I don't need it because I know somebody else makes the same drug product and it'll be just fine. And I've been doing the same thing for years. I've never had a failure. I never hurt anyone. And by the way, the state board didn't ask. Okay, good. Man, everyone loves to blame the state board for not doing the internal evaluation that should have been done by the pharmacy in the first place. But by the way, 95% of you guys already do this. There are 95% of the people out there, and I go into a lot of pharmacies, they ask these questions. They see what's going on. They know what's happening. And saying that you never had a failure is the reason you do method suitability. Years ago, I used to test a product that was penicillin. I had to add to my mixture penicillinase to get rid of the penicillin to make sure I could pick up other organisms that may or may not have been in there. So you had to neutralize what was happening and what was going on. There is a reason method suitability exists. And the pharmacy compounding world makes some pretty unique products. And all of those things, you deserve the right to know that what you've done is safe. And I believe they're safe. But I'll tell you, method suitabilities fail. And alter things have to happen. They don't all pass. But there's other things that go on. And a growth mindset says, wait a minute, I have to do it by my dosage form. And I have to be formula specific. And formula specific is another one of those. Now, there's actually some boards that disagree with what I'm about to say, but I'm having a science discussion here. If I have the highest concentration of a drug and I do a method suitability on that, all the ones beneath it pass. Thank you very much. Because I'm worried about that active ingredient inhibiting the ability for my media to grow. A growth mindset says, excuse me, that's the right answer. Now, having said that, if there is a regulatory requirement to do all of them, then my growth mindset is also going to say, okay, no proclamations. I must follow my regulatory compliance issues and I will do so. But from a science perspective, I know it's the highest concentration. I also know something else that gets confused. And the confusion is USP 71 says very specifically, if in fact you can filter a product, you must use membrane filtration to do it. If you can't, then you use direct inoculation because it's non-filterable. Now, there are many times, all because of cost, it is much more expensive to do membrane filtration than direct inoculation, that those are substituted as equals. They may well be equals, but it's not correct from a regulatory perspective. Now, I happen to know in some of the things that we do that they're not always equal. Membrane filtration does a very nice job. It's a very good job. And if you were, I was going to say old, aged like I am, you would know that years ago, that if you did membrane filtration, you only had to incubate it 7 days. And immersion was 14 days. There's all sorts of things that happen over time that we get lost with. But growth mindset says whatever you're doing, understand why you're doing it and what it means. And the sample size is a probability based on batch size. Please understand, 99% of the products that we all make are sterile, but here's the but. If they're contaminated, it's usually a small percentage, or it's all contaminated, forget about it, something else had a breakdown. So every time we see or we hear, well, I failed the sterility test, I sent it to another lab, I passed, it must have been a lab error. Go with no on that. It's no different than you're going into Las Vegas and you're putting money on 00 on the roulette wheel. It doesn't come up every time. It is a probability function, it's a distribution function, and that's why that doesn't work. So let's talk about OOSs. OOSs is really some of my favorite with a growth mindset versus a fixed mindset. Literally, there are individual decisions that can be made to just throw it away and start over. And by the way, even though it's a fixed mindset, I agree with that, but I don't stop my investigation. If it's a low value, low quality, is just going to be a problem, you can throw it away if you want to. But what I'm going to do is continue the process. And I'm not going to just look at that lot. And I'm sure as heck not going to test in compliance. And I'm going to assume, it's my fault. I want to prove that I was wrong, not that I was right. When you look at a growth mindset for OOSs across the board, I don't care if it's a sterility test, a potency, environmental monitoring. There's my favorite fish. And boy, if you knew the age of that fish is older than I am. Man method material machine, and we always throw in Mother Nature because here in Texas, and I'm in Houston, we get a lot of rain, hurricanes, and you just never know what's going to go on. But when you do an OOS, you have to make sure that you get your arms around when was it good? What changed between good and bad? You ask those type of questions. You don't look at one lot, you look at multiple lots. You don't look at just one drug API. If it's the same dosage form and the same process, look at all of them. You have to look at the similar products. You have to look at the ones that are made the same way. You have to do that. That's a growth mindset. You will see item after item where you'll get notice in warning letters and 483s and Board of Pharmacies saying, well, it was an insufficient OOS. Usually what that means is that you look too much at what happened as opposed to what could have happened everywhere else. Please keep that in mind from that perspective, because I must tell you that will help you become more efficient. It will mitigate risk, and it will help you understand your process even better. Please do that. Okay. Engineers love equipment. I love equipment. I like something, I take it home, I plug it, I want it, I plugged it in, I turned it on, I say, damn, why didn't it work? Oops, sorry. Why didn't this work? I don't get it. Well, that's a fixed mindset. Boy, and I've got it when it comes to that. Engineers, on the other hand, that are quality engineers and GMP people, they fill out more forms on a piece of equipment and it's still not always enough. Now, I put one down here, and this is a form we use here at Eagle, okay? And what I'm trying to tell you is when you buy a piece of equipment, I'm not telling you not to believe the sales rep. I don't think I am. But what I'm telling you is ask them some questions. Ask some technical people. But the first thing you need to do is you need to write down your user requirements to know exactly what you want, exactly where it goes. You need to know what it is. And you need to ask yourself the question, will this accomplish without causing a problem? Many pieces of equipment cause problems, don't they? We didn't plan on doing it, but I forgot to calibrate, or my balance didn't get this, or my incubator didn't do that. You have to worry about things in our world that are called IQ, OQ, PQ, installation qualification, operational qualification, performance qualification. But a performance qualification says an oven turns on. It doesn't tell you what you have to set that temperature to bake your cookies. It's no different in the restaurant world. It's no different in food processing. All of these things are processes. All of these things require not only getting them set up right for the intended's use, but to make sure that they're maintained, to make sure that if the process changes, now I'm putting in 2 cakes versus 1 cake. You already know the process is different, right? So all of these things should be discussed, understood, and absolutely covered in your SOPs and procedures. Because I think many times we lose track of the fact that I set my temperature and I did a full load and everything is good. Well, that's not always true, because if you're using a full load and you start it cold, it will take that oven for dry heat, for instance, what I'm thinking in my mind now, it will take that a lot longer to come up to temperature, which means you're going to get a lot more integrated heat, lethality, and destruction. That if you put one thing in there, it comes up immediately, doesn't sit there long enough to get depyrogenated or sterilized. Think through things, and please, please investigate what you don't know. Like I said before, I know Zippo about pharmacy. If a pharmacy question comes up and dosage forms, I am calling up, Gus and AJ and Megan. Somebody tell me what I'm supposed to do, because I don't know. I don't know. And even if I think I know it, I get even more nervous. So please make sure that growth mindsets are always a process of knowledge, a process of education, a process of understanding. In sanitary conditions, here's a picture of an obvious thing, right? There we go. My place is clean. I'm clean. I use a filter. That's what this individual told me, and I'm looking at that, okay? And the answer was, well, that's okay. That's my 795 area. I didn't know dirt was good in any area in a pharmacy. I don't like it. When I walk into a pharmacy, when I walk into it, my mindset is all about, are there cobwebs? Are there bugs? I can't tell you how many times we go into a pharmacy, or I go into a pharmacy just to say hello, or a 503B, guys, and there are bugs in the lights, but it's outside the controlled area, right? So it's like in the hallway, and the person telling me what doesn't count there. Stop. Of course it counts there. Of course all these things count there. You cannot have 2 sets of requirements for common sense. You cannot do that. Stop. I've got kids. You should not do that. Okay? So now the next one, I just want you to know, we've heard so much about these things of using inactive and active ingredients that have higher levels of impurities. I used to talk about the Piggly Wiggly olive oil. I apologize to my friends in New Jersey that go to Piggly Wiggly that were being used in injectables. Well, it looks like, and these are 6 weeks ago in a pharmacy using Mazola. Just stop. Just stop. There isn't anything that I can say, but this makes no sense. And this, by the way, hurts our industry, because there is no excuse for this. All right, so listen, when we get into this, I want you to know that engaging in aseptic processing, wearing gowns at the wrong time or the right time, exposed skin. What I want you guys to do, what I want everybody to do is to think about what you're doing and the reason you're doing it. Engage in critical thinking. Common sense has got to prevail somewhere, right? And I will thank Megan, who put this slide together for me. She's the classic, if it looks like a duck, sounds like a duck, must be a duck. So what I'm telling you guys, leave your comfort zone, step out of your shoes, right? Accept the challenge of thinking about what you're doing, get your mindset going, and I thank you very much. And this is the book. It's an excellent book. It's good for family. It's good for everybody. I obviously sent it to all my kids, and actually I bought one for everybody in my company. So please enjoy. Are there any questions?
Great. Well, thank you so much for joining this live Q&A. We have just a couple minutes, but a reminder to attendees, if you would like to ask a question, click on the button on the bottom right-hand side of your screen. So we'll jump right into it. For Dr. Caputo, the first question is, I am a 503A pharmacy. Do I have to wear sterile garments in the clean room?
All right, so that's a two-part question. One, the regulatory requirements requiring you to do that, and I don't believe in 797 you have to do that right now, but I absolutely would. Absolutely would.
All right. The next question is, can an outsourcing facility use an EP or BP grade if a USP grade is not available?
Not only am I not a pharmacist, I'm not a lawyer. I do believe, though, that is some question that's being discussed, and I believe the answer is that you cannot, but I do not know that for a fact. I would ask you to talk to a lawyer.
We have a potential New Jersey resident who writes in and says that there are no Piggly Wigglies in New Jersey.
Oh, no.
All right, the next content question is, do I have to perform all of the tests listed on the USP monograph, or can I use the certificate of analysis from the manufacturer?
From my perspective, when you are getting a qualified C of A from a manufacturer, and if you're a 503A pharmacy, I believe that you're good to go. I also understand that if you're doing a potency test and an activity like that, you're already going to see if it's the right thing at the right time. However, the one thing that you need to be careful of is read the C of A, because the potency is not always 100%. Sometimes it's 98, it's 96, it's 101, it's 102. You have to correct for that. There have been some situations where they've missed water content, they've missed all sort of activities to look at it. A 503B, however, has got to do a bit more work on approving their vendors, a bit more work in getting it done. And I think that is something that we should negotiate and talk about, because the answer to that one actually is, it depends.
Great, thank you. The next question is, what is your recommendation for fingertip surface testing? USP 797 states you only have to do fingertip testing every 6 months.
Microbiologist, we run a lab. We do sterility testing every day. We do fingertips and surface counts every day, every batch.
Great. Well, thank you so much. I know that we are just about out of time. So I do want to thank everyone again for joining and submitting questions. And a reminder, you'll be able to view this recorded session on demand starting tomorrow. And the slide deck will be available in the delegate bag on the portal. But thank you again. A special thank you to Dr. Caputo. And I'll hand it over to you for any closing statements in this last minute here.
Good. Thank you. I want to tell everybody, I think that the compounding industry has come a long way in a time and an era that hasn't been easy. Think about all these activities and changes that are going on, COVID, people being sick. We lost friends. I hope none of you lost friends, but many of us did. We all know people that were sick, and we still had to do the job to get it done, because we're about patient safety. We're about taking care of people. So we have to do that, frankly, with the right philosophy, right? And our right philosophy is that we're there for you. We are there for the patient. We are there to make sure that we can do whatever we can do. And frankly, when we make a mistake, stand up, dust yourself off, and move forward, because it's not the end of the world. Take care, and thank you for your time. I appreciate it.